Because of my carelessness, I cancelled my presentation at APBC 2018. I'm so sorry for organizing committee. So here I upload my presentation planned to be presented in APBC 2018. My talk of title of my talk is tensor decomposition based and principal component analysis based and supervised feature extraction applied to the G expression and maturation profiles in the brains of social insects with multiple cases. My name is Taguchi from Department of Physics, Chuo University, Tokyo, Japan. Generally, phenotypes are not always directly related to genotypes. For example, adult and child share same genotypes, but its phenotypes are completely different. So another typical example is male and female. For human beings, the male or female is decided by the genome, but for example, for other species, fishes, gender or sex are not decided based upon genome at other experimental environmental conditions. And same genome with distinct phenotypes is possible. And such a typical example is social insects with caste. The many well-known social insects, ants, bees, termites, and this is typically a queen and workers, and of course they are same species, they share same genome, but their phenotypes completely different. So then in this case, epigenetics is possibly candidate to control these different phenotypes. And we would like to analyze some data set in order to understand what causes the distinction between workers and queens in these social insects with caste. And first, we downloaded the gene exposure and measured profiles from this GSE 59525 gene expression omissive dataset. It includes two species, this is bee and ant. And for gene expressions, for bees, 4 queens and 6 workers are included, and for the ants, 7 queens and 6 workers are included. So measured profiles for both species, 1 control and 3 queens and 3 workers are included. The purpose is identification of genes associated with aberrant gene expression and measured profiles between queens and workers simultaneously, because if the epigenetics can control the phenotypes distinct between queens and workers, these genes should be governed by some epigenetic effect, for example, methylation. The method we used is principal component analysis based unsupervised feature extraction, which I have invented a few years ago. And what is PCA based unsupervised FE? So here, typically for gene expression or maturation profiles in this presented the matrix. So the number of samples are very small, while the number of genes is huge. So this is typical large P small n problem. And among huge number of genes, only limited number of genes are distinct between samples. In this case, these samples composed of 2 classes, the workers and queens. Then principal component analysis is applied to the Epsilon matrix so as to embed each gene in low-dimensional space. In this example, we have embedded the genes into 2-dimensional space. And then, if the limited number of genes whose expression distinct between classes are placed apart from other genes, because this axis represents the distinction between classes, as you can see in this display. This is a typical example using a synthetic dataset. There are 100 features. This corresponds to genes, and the 2 classes. This correspond to queens and workers. And these two classes, gene expression profiles are drawn from the Gaussian distribution having distinct mean values. For this part, gene expression are drawn from completely mixed, thus no distinction between two classes. We embed these 100 features into two-dimensional space using PCA. As you can see, the red triangular, which corresponds to these 10 features, are located apart from others. And blue cross is top 10 outliers. And then, as you can see, 9 among 10 genes are correctly selected, simply employing the top 10 outliers. The important point is getting this embedding is without labeling information. We simply apply principal analysis, this matrix, without using labeling information. We can get this embedding and we can identify genes which are distinct between two classes. Thus, this method is completely unsupervised without any labeling. Of course, it depends upon the distinction between two distributions. So this distribution difference means is as large as 2, so accuracy is 90%, but if the mean value is decreased to 1, then the accuracy is reduced to only half. But still, it is significant because only 10% is true positives. Then we applied this method, PCA-based unsupervised FE, to real dataset. And first, PC loadings attribute to samples, in this case workers, queens, and then if the PC loadings distinct between the workers classes, we can select these PC loadings to select genes. And corresponding PC scores taken from the PC loadings, we can select genes using p-values obtained by assuming a chi-square distribution for distributions of genes. And this is an example of methylations. So for P's, we can find first PC loadings which attribute to the samples exhibit the distinction between controls and queens and workers. And here we can draw the histogram of distribution of the PC scores which attribute the genes. And here the origin, the red part are selected because this is completely different distribution from others. And then for ant, we can have similar PC loadings. First one also distinction between control and queens workers, and first PC scores has this distribution. Only limited number of genes apart from the origin where most of the genes are located. Then we have selected this red part for genes which should exhibit the distinction between control and worker and queens. And next, we'd like to check if the selected genes are distinction between queens and workers. So this is principally a distinction between control and queens and workers. As you can see, there's little difference between the workers and queens. So it is possible these genes are distinctly expressed between the queens and workers. And here, this is an example of test. So using a t-test and Wilcox test and Kolmogorov-Smirnov test, we can find for most of the cases for G, B's and ants, these genes are distinct between workers and queens. So we can successfully identify genes whose methylation are distinct between queens and workers. And for gene expression, we can have similar result for third PC loadings and fourth PC loadings. For this is for bees and this is for ants. we can have distinction between queens and workers, and the red part are selected using third PC scores, fourth PC scores. And it is also selected by using a p-value, adjusted p-value is less than 0.01. Then we can apply the success test again to check if these genes are distinct between the queens and workers. Again, excluding one case where we use Wilcox rank sum test. For other cases, or 3 statistical tests identifies the distinction between the workers and queens for 2 species. Thus, basically, we have successfully identified the genes which are expressed distinctly between queens and workers. And finally, in order to check our method can work for other data set, we have downloaded another data set taken from these references and for B's. And it includes not only workers, queens, other 2 cases. And we can find third PC loadings are distinct between 4 classes, and genes are selected using third PC scores. And the blue part distinction between in this order, and pink part distinction between following these orders. Then we would like to check if the selected genes are distinct between the 4 classes. And then we have compared each class and others, workers and others, queer and versus others, and foundless others, and color and others. And genes selected blue part and the pink part are tested independently. As you can see, this asterisk means the p-value is very small, so less than the numerical calculations. And as you can see, for most cases, at least one of 3 statistical tests can identify distinction between these differences. Only expression is distinction between queens and others, but still this test can detect. And only founder less and others on this plus this part cannot have distinction between the founders and others. But for almost all cases, at least one statistical test can identify the distinction between one class and others. Thus, we have successfully identified genes whose expression are distinctly between most of one of classes and others. But unfortunately, selected genes by gene expression methylation are not significantly overlapped. Then we have used another method. This is tensor decomposition. The tensor decomposition is some kind of extension of principal component analysis. And if there is 3-mode tensor, we can decompose into smaller core tensor and small matrix. And using these matrices, we can find genes which is distinctly expressed or mutated between classes. So the situation is like this. So here the methylation and the gene expression, the genes. This kind of tensor has been made use of mathematical product of gene expression matrix and methylation matrix. Then the tensor decomposition applies and this part. So this is singular value vector attributed to methylation. This is a singular value vector attributed to gene expression selected. And this corresponds to PC longx and PCA, and we can identify which PC longx, which singular value vectors are distinct between, for example, for methylation, the control, and queen and workers, and for gene expression, the work and queen. Then we have selected which singular value vector attributed to genes corresponding to the selected gene expression singular value vector attributed to methylation gene expression, and applying statistical test using chi-squared distribution. Then we can compute the p-values for each genes, and as we have done in PCA, we can select the genes which is very small p-values. This is the result. For B's, so for singular value vectors attributed to samples has distinction between control and workers and queens. And for singular value vectors attributed to gene expression, we can find the distinction between queens and workers. And this is the first and the third singular value vectors. And the core tensor having first singular value vectors attributed to the methylation, and the third singular value vector attributed to gene expression have larger values for 9th, 10th singular values attributed to genes. Then we have decided to these two singular vectors to select the genes, the red part. And then for ants, we can do similar things, and we have found singular vector attributed to the first singular vector attributed to gene methylation, distinct between control and workers and queens, and fifth singular vector attributed to the gene expression, distinct between the queens and workers, and core tensor attributed to the first singular value vector of methylation, and fifth singular vector of gene expression, larger values when first gene singular value vector attributed to gene expression is 11th large values. So we have decided to apply using this singular vector to select genes. And we have again checked this select genes distinct between queens and workers for both gene expression methylation. For bees, we can successfully identify genes which are distinctly expressed methylation between the workers, queens, but for ants, selected genes are not significantly distinct, methylated. Only gene expression are distinct. So we could not fully successful, but at least we can find genes which is expressed distinctly between workers, queens, as well as methylated between queen and workers for bees. We have also validated our selected genes using a Go enrichment analysis, and as you can see, because of lack of time, I cannot explain the details. So as you can see, our selected genes has limited some Go terms are enriched, but the original study, where the authors investigated this dataset, there are no enrichment. We also check the selected genes by second GX profiles, and we can find significant enrichment Go terms. This is result for ants. Again, our method can find significant enrichment of Go terms, but as I have told you, the TD-based method is not successful, and there's no Go enrichment. And in the original study, they have also selected enriched Go terms, but our Go terms are more biologically sound. Conclusion is using PCA-based antibody FE, we identified genes associated with distinct methylation gene expression between queens and workers for two social insect species with case. And in addition to this, using TD-based antibody FE, we identified genes associated with distinct methylation gene expressions simultaneously between queens and workers for one of two social insects. So this is bees. And various Go terms are enriched in select genes. Thank you very much for your interest.